Okay. Hi. Let me get my glasses. Oops. Sorry about that. All right. So this is going to be the first screencast about cellular respiration and photosynthesis. And first, we're going to talk about the structure of mitochondria and chloroplasts because their structure really determines, again, their function. All right. And we know that they both came about through the endosymbiont theory. And on the last test, there was some confusion about what that actually means. And the endosymbiont theory is the theory that describes how mitochondria and chloroplasts evolved from prokaryotic cells, right? So let me go ahead and share my screen. And we'll get going. Okay. So I'll post this obviously online, both the PowerPoint slides and the screencast. All right, so we are talking about mitochondria and chloroplasts. And as we know, in the mitochondria, whoa, that's a thick pen. This is where we're gonna see cellular respiration, right? And in the chloroplast, that's where we're gonna see photosynthesis, right? And remember, these are kind of reciprocal reactions where cellular respiration gives off CO2 and water, right? And those are needed for photosynthesis. And photosynthesis gives off oxygen and glucose, C6H12O6. And those are necessary to run cellular respiration. So they really go hand in hand. So they have a lot of similarities, mitochondria and chloroplasts, right? So we talked already about how they both evolved from prokaryotic cells, right? And that's described by Lynn Margulis's endosymbiont theory. They both are double membraned. They contain their own DNA and ribosomes. And remember, their DNA looks like prokaryotic DNA, right? Which is one singular circular chromosome. And remember, when we're talking about DNA, that means that it is double-stranded, right? They both have very important enzymes and coenzymes, which we're going to learn more about in this section. And something that's going to be really important is that they both build up electrochemical proton gradients across their membranes, right? And we're going to then see how those proton gradients, the energy stored in those electrochemical gradients, will power the creation of ATP from ADP and PI, all right? So remember that when we're talking about building up an electrochemical gradient, we're going to see that these protons are going to be shoved in against their concentration gradient to create an electrochemical gradient. And then we are going to watch how things can flow down their electrochemical gradient in order to create ATP, right? So in that process, we're going to see that ADP is put together with inorganic phosphate to make ATP. All right, sorry that the slides get a little funky when I put them on my iPad. So something to note is that mitochondria is in all eukaryotic cells, right? There's a few random exceptions like red blood cells. No, red blood cells have them. I'm sure I think all cells. I take that back. All cells that I can think about all eukaryotic cells have a mitochondria, right? And that's kind of shows you how important they are for eukaryotic cells. Remember that prokaryotic cells have no membrane-bound organelles, right? So they have no mitochondria, no Golgi, no nucleus, etc. So I'm gonna just move my little face over down here. Okay, so mitochondria are in all eukaryotic cells, plants and animals, right? So there's a big misconception that plant cells, because they make their own food, they don't need mitochondria. But no, they make their own food and then the mitochondria uses that food, right? Whereas humans and other animal cells, we have to eat our food. All right, so the whole goal of mitochondria is to take in nutrients, and this we're going to be really looking primarily at glucose, C6H12O6, And we're going to see how that gets broken down to give the formation of ATP, right? And we know that as you break down glucose, we're going to see CO2 that leaves as waste. We're also going to see some water production. Okay, so the structure of the mitochondria. So there is an outer membrane, right, shown here in this kind of burnt orange color. Then we have an inner membrane, all right, and that inner membrane is folded into these really crazy folds called cristae. All right, so those are the inner membrane folds, and that increases the surface area, and meaning that allows for more regions where ATP can be produced, right? So we're increasing the surface area so that we can increase the energy production, right? So on those inner membranes, we are going to see that they have carrier proteins where, I'm just going to draw some here. We're going to see enzymes. We don't have to know what they are really yet, but there's enzymes. Of course, I drew it right where my face was. Sorry. We're going to see that there's enzymes on that inner membrane, all right? And that those are really going to be very important for kind of creating gradients and for moving things along, all right? So those membrane proteins are on the inner membrane, and they're enzymes that are really important in moving things along and for creating the proton gradient that I told you about. Okay, so the inner membrane is folded into these cristae to increase surface area. The inside of the mitochondria, kind of shown here with this blue watery stuff, that is the matrix, and that is the most internal part of the mitochondria. And it is also going to hold a lot of enzymes. So we're also going to see that the matrix has enzymes, and that are really important for, you may have heard this before, the Krebs cycle, sometimes called the citric acid cycle. And then there's another really important reaction in there, which I'm not going to talk about yet, called pyruvate oxidation, or the link reaction, right? And then lastly is that there is this space between the two membranes, right? So I'm going to actually color that in light green here. So there's this space between the outer membranes and the inner membrane, right? And that is called the intermembrane space, right? And what we're going to see is that the intermembrane space is where the hydrogen electrochemical gradient is going to build up, right? That is where we're going to see the hydrogen electrochemical gradient build up between the inner membrane and the outer membrane, and it's called the intermembrane space. All right, so photosynthesis it obviously occurs in plants, and in particular, so I'm trying to move my face around, in particular, it occurs in a very specific place on leaves. So plants, first off, are green, and that has to do with that they have this pigment called chlorophyll, right? And in particular, chlorophyll A's and B is the pigment that is going to absorb the light energy from the sun, right? And then we are going to see how photosynthesis can then capture that light energy that has been absorbed by chlorophyll and use it, right? So photosynthesis occurs in the green parts of plants because that's where there's chlorophyll. Most of the time, it is going to occur in leaves or leaf cells. Sorry, not leaf cells, in leaves. And if you look at the cross section of a leaf, you can see here, and it's kind of blown up. If you look at the cross section, they have we're going to see that there's a cuticle on top, and that helps prevent water loss. So that's a cuticle. Then we have this meaty region of the plant cell, which is called the mesophyll, right? So the mesophyll is where we're going to see most of photosynthesis taking place. On the underside of cells, we have these little tiny openings that are called stomata, and that is for gas exchange, right? So the stomata are for gas exchange. Obviously, plants are going to take in carbon dioxide, and they're going to give off oxygen as waste, which is awesome because we need oxygen. as one of the reactants for cellular respiration, right? So in a single mesophyll cell, which is shown here, they have about, you don't have to know this number, but 30 to 40 chloroplasts, right? So a chloroplast is an organelle in the plant cell that has many structures, which we're going to talk about in the next slide, all right? So the chloroplast and, again, the mitochondria, those were the two organelles that the endosymbiont theory describes their evolution from a prokaryotic cell. And that's like the evidence, remember, double membraned, they have their DNA, they own DNA, they grow and divide independently within a cell, they have DNA and free-floating ribosomes, etc. All right, so let's look more specifically at chloroplast structure, all right? So chloroplasts, again, these are only in plant and algae cells. They help turn sunlight energy, so they capture the energy from the sunlight through that pigment chlorophyll, and they turn it into glucose that can then be used by the cell to break it down to create ATP, right? So cellular respiration. So in plant cells, they use photosynthesis to make their own glucose, and then cellular respiration turns that glucose into ATP, right? Whereas animal cells or animals, we have to eat our food to get glucose, which then our mitochondria can then use to make ATP. Plants, they make their own sugar, and then they use that to break that, they break that down in cellular respiration. Okay, so let's look at the structure. So chloroplasts have an outer membrane and an inner membrane, just like a mitochondria, right? But chloroplasts are a little more complicated in structure, and that's because inside the inner membrane, they have what are called thylakoids, right? These are these thin pancake-like structures, right? I always think of them that they look like little pancakes. They are membranes, and that is where you have the chlorophyll, right? So when the sun comes in, they're going to hit these thylakoid membranes, and the thylakoids are going to be really important for photosynthesis, and in particular for the light-dependent reactions of photosynthesis, because that's where the chlorophyll is, right? So those are the thylakoids or thylakoid membranes, I often call them. And just to make things even more complicated, the thylakoids, when they're in a stack of pancakes, that is called the granum or a grana, right? So here are the thylakoid membranes, right? Together in a stack, they make a granum, right? So each one is kind of a thylakoid membrane. And together in a stack, they're called a grana or a granum, right? Grana is plural. So inside the chloroplast, there's lots of grana, right? Each grana has lots of thylakoids. And then the kind of cytoplasm or the cytosol within the chloroplast is called the stroma, all right? So that's, you know, all this region around the thylakoids is filled with fluid, right? And that is the stroma. And in the stroma, we're going to see that that's really important for the light independent reactions. And we'll talk about all of those later, right? So I just want to mention the last thing here is about these proton electrochemical gradients that I told you about that are really important for making ATP. And they're important for making ATP in both mitochondria and in chloroplasts, right? So in the mitochondria, the hydrogens are pumped into the intermembrane space, and that is kind of shown here with that darker beige color, right? So they're going to be pumped into that space. So if we zoom in, we're going to see that hydrogens are going to be pumped into that space, right, from the matrix. And then we're going to see that those hydrogens can then flow down their concentration gradient through this enzyme called ATP synthase, right? So ATP synthase is this weird molecule that sits on the intermembrane, and hydrogens are going to diffuse down their concentration gradient that was created, and that's how we're going to make ATP, right? So So if you zoom in here on the matrix, so on the mitochondria again, so hydrogens are pumped into that intermembrane space. And then we're gonna have our little ATP synthase, where these hydrogens are gonna go down their concentration gradient. And when they do that, we're going to make ATP, right? So similarly, if we look at chloroplasts, we are going to see that hydrogens are going to be pumped into the thylakoid membranes. They're going to then, that's going to create a electrochemical gradient in the thylakoid membranes. The hydrogens are going to then flow down that concentration gradient through ATP synthase to create ATP. So if we zoom in on a chloroplast here, hydrogens are going to be pumped into thylakoid membranes. On the thylakoid membranes, we're going to have an electrochemical gradient build up. Those H's are then going to go down their concentration gradient through ATP synthase, and we're going to see the production of ATP. Okay, and that is it. So let me stop my sharing. And that's kind of the basic structure of mitochondria and chloroplasts, and kind of an intro to cellular respiration and photosynthesis. All right, chip, chip, cheerio, toodles, poodles.